Hello, my name is Tiaja, and my group will be presenting chapters 2.1 through 2.2, and my group members are Emily, Daniela, and Olayemi. Carbohydrates get their names because they are hydrated carbon or carbon with water. The formula shows that carbon dioxide and water is used to make a carbohydrate. The N in the formula indicates the number of times the formula was repeated. Carbohydrates can be divided into simple, complex, sugar alcohols, and alternative sweeteners. Simple carbohydrates can be divided into two groups, the first being monosaccharides. Monosaccharides contain one sugar. The three monosaccharides are glucose, galactose, and fructose, and they are all 6-carbon sugars. Glucose and galactose are structurally similar, but they both are 6-ring members. The only difference between them is the position of the alcohol group bounded to the carbon. Fructose is a 5-member ring, and as we all know, glucose is a product of photosynthesis and is a major source of energy in our bodies. Fructose is commonly found in fruit and is used commercially in beverages. Galactose is normally found in the disaccharide lactose, which we'll talk about next. Disaccharides are produced from 2 monosaccharides. The most common types are maltose, sucrose, and lactose. Maltose is produced from 2 glucose molecules binding together, and maltose can be found in alcoholic beverages. Sucrose is produced from glucose and sucrose and is a sugar found only in plants. Lactose is a product of galactose and glucose, which is a primary milk sugar. High fructose corn syrup is a cheaper alternative made from corn syrup. It contains 42 or 55% of fructose, and it's also believed to contribute to an increase in obesity in the US. Sugar alcohols are another type of carbohydrate. They are nutritive sweeteners, which mean they do provide calories. The 3 most common sugar alcohols are xylitol, sorbitol, and mannitol. Sugar alcohols are nearly as sweet as sucrose, but provide half the calories. And on the chart, you can see how compared to lactose, sucrose, and fructose, which are typically relatively sweet, the 2 sugar alcohols, isomalt and lacticol, are pretty sweet, but they provide half the calories towards the other sweeteners. Another thing about sugar alcohols is that they don't contribute to tooth decay compared to other types of sugars. And there's a video linked to this presentation that shows the process of tooth decay. Next are alternative sweeteners. These type of carbohydrates are non-nutritive and highly intensive sweeteners. So non-nutritive would mean that they don't provide any calories. It's an alternative to sucrose and other mono and disaccharides. They're developed and they're usually used in 0-calorie or low-calorie sweetening for foods and sugars. Some common types of high-intensity sugars will be aspartame, saccharin, sucralose, stevia, and Lao-Hao-Go fruit extracts, which is commonly known as monk fruit. And you can see in the image the fruit, and then the white substance is the sugar form of the extract, the fruit. And stevia and monk fruit are derived from plants, so they're more natural sweeteners, while the other forms of high intensity sweeteners tend to be synthetic. Next are complex carbohydrates, the first being oligosaccharides. These type of sugars are composed of 3 to 10 sugar units. The most common form of most common types of oligosaccharides are raffinose, sachiose. These type of carbohydrates are found in legumes, onions, broccoli, cabbage, and whole wheat. This type of carbohydrate is known to cause digestive issues because humans don't have the enzyme to digest them. Typically what happens is once oligosaccharides reach the colon, they are fermented by bacteria there, and gas is produced as a byproduct, and this bacteria fermentation can lead to flatulence. And the next type of complex carbohydrates are known as polysaccharides. Polysaccharides are composed of many or more than 10 sugars. The main classes of polysaccharides are starch, glycogen, and fiber. Within starch, there are two forms. Within starch, starch is a storage form of glucose in plants. There are two forms of starch. One is amylose, which is a linear polysaccharide. The other is amylopectin, which is a branched polysaccharides, and you can see the two types of structures. Glycogen is another polysaccharide. It's the storage form of glucose in animals. The structure is highly branched and it contains lots of alpha glycosidic bonds. Fiber is an indigestible matter. Fiber can be classified by their physical properties of fermentable or viscous. Fermentable being whether bacteria in the colon can ferment. These type of fibers include hemicellulose, pectin, beta glucans, and gums. They're typically found in oats, rice, psyllium seeds, soy, and some fruits. And then you'll have non-fermentable or non-viscous, which is insoluble. These include hemicellulose, again, cellulose, and lignin. They're found typically in whole wheat, whole grain cereals, broccoli, and other vegetables.
About proteins. What is a protein? They are made up of small repeating units of amino acids. Proteins contain carbon, hydrogen, oxygen, and the most distinctive factor of them all is the nitrogen. The amino acid signifies that each contains the amino group right here. I'm circling around it. And they also contain a carboxyl group, which is right here. It has the CO and OH. And then the R group, which is right here. They're called the side group, also called the side group. And this feature is what sets amino acids apart. Amino acids and peptide bonds. So peptide bonds are made when two amino acid joins together. And right here, we have this amino acid 1 and amino acid 2. Again, the R group, you see that? You see the carboxyl group right here and the amino group right here. These two are going to join together. And when they join together, they release 1 oxygen and 2 hydrogen, which is H2O, also known as water. When it releases, this process is called, it releases water, and this allows for there to be a bonding, which is what the peptide bonds are called. And when then right here, the green arrow, you see the peptide bond. Now we have a whole new amino acid, which is contained with 2 separate that are now 1. So amino acids can combine, 3 amino acids can combine to be called tripeptides. Then from 3 to 10 amino acids, when they combine, are called oligopeptides, and when they are 10 and more, they're called polypeptides. And we're very familiar with polypeptides. And poly meaning plenty, peptide bonds, so plenty amino peptide bonds. So now we're going to talk about protein synthesis. Protein synthesis has three processes: transcription, translation, and elongation. The book is not going to focus on elongation, but it will focus on post-translation. So these are the three things that I will be going over in this part of the chapter. Transcription occurs in the nucleus. And in the nucleus, as we all know, there is a DNA. And in DNA, there's a template that will create an mRNA. A DNA cannot leave the nucleus, but an mRNA has the ability to leave the nucleus and help the processes of making a protein. So in transcription, the DNA will use its template to create an mRNA, which will then leave the nucleus and go into the cytoplasm right here outside of the nucleus, where mRNA will then act as a template, right? In which this mRNA will go to another RNA called a tRNA. The tRNA is where the mRNA will go, and the tRNA will use the mRNA as a template to create a polypeptide. So it's basically going to make amino acids and form multiple peptide bonds, and it will make a polypeptide. And then in the post-translation area, now we have a polypeptide, and in the post-translation, the protein itself will start to fold in itself, right? It will fold to make a specific shape, and that specific shape is what will help the protein exactly with the DNA. It will help the protein know exactly what to do when it folds on itself. This is what distinguishes this protein from any other protein. And it also differentiates the protein into whatever it needs to be. And then right here, there's an effector molecule that will bind to this newly made protein, and this effector molecule will activate the protein. So now this protein, whatever this protein is made for, maybe it is a kidney protein, or maybe it is a heart pumping protein, this active effective molecule will allow this protein to begin the processes that it's supposed to make.
First off, I want to start off by talking about protein structure. The primary protein structure is a linear polypeptide chain. The secondary structure of a protein is when hydrogen bonds between amino acids form in the same polypeptide chain, causing the formation of either an alpha helical structure or a beta pleated sheet. The tertiary structure is an attraction between different amino acids of a polypeptide chain. And the quaternary structure is multiple polypeptide chains that are bonded together to form a larger molecule. An example of this would be hemoglobin. Now I want to talk to you about protein function. The structural function of a protein helps to serve as scaffolding for the body. An example of this would be collagen, which serves as structure for different tissues. Then we have hormonal function, wherein a compound is produced in one tissue and released into circulation to affect a different organ. Then we have immune function, where antibodies are proteins that recognize antigens and bind to them to inactivate them. For fluid balance, proteins maintain balance between different fluids in the plasma and interstitial fluid. Buffers prevent the pH from getting too high or too low in the acid-base balance. And then for transport, proteins move molecules through the circulation across membranes, such as hemoglobin, and how it transports oxygen. Now I want to talk to you about the different types of amino acids. So there are three different types: essential, non-essential, and conditionally essential. Essential amino acids are not synthesized in the body, therefore we must consume them. Examples of this would be histidine, leucine, lysine. Non-essential amino acids are made naturally in the body, so we don't need to consume them, such as alanine, serine, asparagine, and then conditionally essential amino acids become essential in the case of certain diseases.
Chapters 2.26 to 2.28. So 2.26 is just structures of amino acids that were talked about throughout the chapter. And we didn't feel like we needed to put them all on a slide. They're on page 93 of the textbook. So if you want to check those out, you're more than welcome. 2.27, however, is about protein quality. Protein quality is classified in two ways. There's incomplete and complete proteins. Complete proteins provide enough of the 9 amino acids that are essential to our diets. And these are typically animal proteins like chicken, beef, pork, whereas incomplete proteins don't provide enough of those amino acids. And they often have a limiting essential amino acid. So these are the amino acids that are not provided in a sufficient enough amount to make the protein a complete protein. However, incomplete proteins can be paired together and called complementary proteins. These are proteins that supply each other with the amino acids that are missing from the other protein. Now, these things don't have to be eaten at the same time. They just need to be eaten within the same day. So you could do 3 different complementary amino acid or proteins throughout your day among your 3 meals or snacks and stuff like that. There is a puzzle analogy in the textbook that says one person has 2 puzzle pieces and another person has 2 puzzle pieces, and the total puzzle has 4 pieces, but each person on their own doesn't have the whole puzzle. So when you bring this together, you do get a whole puzzle. And that's seen with the complementary amino acids and proteins in our diet. There's a website that you can visit. The link is in the textbook, and it helps you determine the protein quality of your food. And it helps create a balanced diet by telling you if the proteins you're eating are complete or incomplete. And when you have an incomplete protein, it'll help you pair that protein with other proteins that are its complementary. These are all measures of a protein quality. These equations can be found in your textbook, and they're all very important in determining whether a protein has a sufficient amount of the amino acids that we need in our diets. However, the FDA has recently recommended that this measure down here, which the full name is Protein Digestibility Corrected Amino Acid Score, should be replaced with a measure called Digestible Indispensable Amino Acid Score, which measures the amount of the digestible amino acids in a protein in 1 gram of the protein, as opposed to measuring the digestibility of the entire protein itself. Now, when we don't eat protein in enough quantities, we have what's called malnutrition. And typically protein deficiencies don't really occur on their own. It's normally paired with a poor energy intake as well. And we get protein energy malnutrition, which is insufficient energy and nutrition intake. There are 2 different types of this malnutrition. One is called kwashiorkor, and it's a disease that the first child gets when a new child comes along, is what it means. It typically is accompanied by a swollen abdomen and very skinny bone structures. And the protein level is low, but the energy intake is pretty high. It's a decent amount of energy, but they're just not getting enough protein in their diet. The other disease is called marasmus, and it means to waste away. These are very skinny people. They have very minimal energy and protein intake, and it looks like they're just wasting away. Thank you for watching all of our recordings about protein and carbohydrates. I hope you guys got a lot out of these, and